Good afternoon, everyone, and welcome to the Alberta Transplant Institute's research seminar. My name is Jason Acker. I'm a professor here in the Department of Lab Medicine and Pathology and chair of the research committee for the Alberta Transplant Institute. And it's my pleasure to introduce the speaker today. Before we begin, I would just like to start with a land acknowledgement. We acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, languages, and cultures of the First Nation, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. Thank you to Paladin, Astellas, and AstraZeneca for their support of the ATI seminar series. A reminder to all of those attending locally that the ATI Research Day is happening on May 15th. It will be delivered in a hybrid format, so even though those of you that are calling in remotely, there is an opportunity to participate in the Alberta Transplant Institute's Research Day. The in-person part will be at the Bernard Stell Hall here at the University of Alberta Hospital. Registration information can be found in the chat, so please, if you haven't registered, we would love to know that you're coming and attending, so please complete the registration. So with that introductory comments, it's my pleasure to introduce Dr. Janet Elliott, who is a University of Alberta Distinguished Professor and Canada Research Chair in Thermodynamics in the Department of Chemical Materials Engineering here at the University of Alberta. Dr. Janet Elliott is also an adjunct professor in the Department of Lab Medicine and Pathology and is the editor-in-chief of the Society for Cryobiology's journal, Cryobiology. Dr. Elliott's research has been recognized both nationally and internationally in science and engineering by numerous organizations, including the American Chemical Society, Langmuir Lectureship Award. Dr. Elliott is a fellow of the Canadian Academy of Engineering, a fellow of the Royal Society of Canada, fellowships in the American Institute of Medical and Biological Engineering, fellowship in the Society for Cryobiology, a fellowship in the Chemical Institutes of Canada, as well as a fellowship in Engineers Canada. In addition to all of the numerous international and national awards, Dr. Elliott has also been recognized locally for her teaching and research, most recently with Faculty of Engineering Graduate Teaching Award, as well as other awards from APEGA and other organizations from here in Alberta. And as one student has eloquently put it, and I probably know who that student is, she could convince rocks to study thermodynamics. So with that, I'd like to welcome Dr. Elliott, who will give her seminar on cryopreservation of cells, monolayers, and tissues.
I'm very happy to be able to give this seminar here today, both to our friends from the Society for Cryobiology and from the Alberta Transplant Institute. So it's great to have this joint webinar. And I'm going to be talking today about my group's research on cryopreservation of cells, monolayers, and tissues. And I'm just trying to see how I'm going to do that. Well, these are some of our funders here, and without further ado, I will give my ATI Declaration of Conflicts of Interest slide. I receive an honorarium for Society for Cryobiology in my role as editor-in-chief of the journal Cryobiology. I serve on the following boards that are unpaid positions. I have patents filed and allowed related to the research I'm talking about today. I will not discuss off-label use of drugs or devices, but I will discuss the results of research that are aimed at future human applications that have not been used in human treatment yet. I just want to mention before I get started that this research was done by lots of people, my graduate students, my research staff, and collaborating professors, and their names are given as co-authors of papers cited on the relevant slides. Some images are copyright by the individuals that made them or by the journals in which they are published as documented in the journal paper citations. And the research on animal and human cell and tissue samples that I'm going to present today was approved by the University of Alberta Research Ethics Board, and the animal samples came from the waste products of meat production. So I know that members of the Society for Cryobiology are very familiar with cryobiology, but some people from the Alberta Transplant Institute will be less familiar with cryobiology, and I'm just going to start out with an introduction to cryobiology in general. Cryobiology is the study of the effects of low temperature on biological systems with a major something just happened where I cannot, oh, there we go. With the major application being cryopreserving cells and tissues for transplantation or research. Many things are routinely cryopreserved: blood, stem cells, sperm, heart valves, skin, bone, tendons, ligaments, commercially available culture cells. Every time you do some biological research and you purchase some cells, they're going to arrive to you in a cryopreserved status. But if cells and tissues cannot be cryopreserved, they're typically not available for purchase for research or not available for widespread clinical use. And in bringing the typical cell therapy to market, some people may not be aware that 15 to 20% of the resources are actually required to develop the cryopreservation procedures. And cryopreserving tissues is even much more challenging than cryopreserving individual cells. And preservation has been recognized as a key bottleneck featuring the, facing the entire industry of tissue engineering being lab-grown tissue constructs that would be used for transplantation. So in order to be used, those things will need to have some way of cryopreserving them, storing them, and transporting them. So there are two different ways to cryopreserve cells. One is classical cryopreservation and one is vitrification. So for cells in suspension, we would normally add 5 to 10% by weight of cryoprotectants, nucleate ice just below 0 degrees Celsius, and have controlled cooling in the presence of ice. And if you do that, then you need to warm fast, but it's not too critical. Here's a picture taken by Loxley McGann of some cells happily coexisting with some ice that's outside of the cells. So this would be classical cryopreservation. On the other hand, for pieces of tissue and larger biological samples, this is just a picture of a little bit of bone with a little bit of cartilage on top. We don't want ice inside the cells, and we also don't want ice outside the cells. So another way to cryopreserve things are with vitrification. So this is using 50% or more by weight of cryoprotectant, cooling so fast that you outrun ice formation and there's no ice present either inside or outside of cells. And if you use this method, then you need to warm very fast so ice doesn't form on the way back. Here is a picture from our research in my joint research group with orthopedic surgeon Nader Jama. It's taken by our PhD student, Kezu Wu. And this is just a partial knee, some bone with cartilage on top of it. This is actually solid and at low temperature, but it's clear you can see through it because there's no ice crystals. It's vitrified. So what happens when we cool cell down? Here's a picture of a cell at room temperature. When you cool it down, we first form ice outside of cells. There's a number of reasons why ice first forms outside of cells. And what happens next depends on the cooling rate. If we cool cells slowly, as we get to lower and lower temperatures, we form more and more ice, and this surrounding solution gets concentrated. And water then leaves the cell to kind of equilibrate with this more concentrated solution, and we get these really shrunken, dehydrated cells. And if you live in this state for too long, eventually that's lethal to cells. And we call this slow cooling injury or solute effects injury. On the other hand, if you cool cells rapidly, so rapidly that the water doesn't have time to leave the cells, then eventually you're going to have some supercooled water and nucleation of ice inside. And this ice inside invariably, especially for cells in suspension, is lethal for cells. And we call this intracellular ice injury or rapid cooling injury. So the game of cryobiology is to find some intermediate cooling rate, which allows you to avoid the slow cooling injury and avoid the rapid cooling injury. And what is slow and what is fast is different for different cell types because different cells have different membrane permeabilities to water. And actually, for almost all cell types, the slow cooling injury overlaps the rapid cooling injury, and there would be no rate at which you could cool them and still have cells alive at the end. So we add some cryoprotectants, and they change the freezing point of the solution, and they change how much ice is formed at each temperature, and they allow us to open up a window and find some path that avoids the slow cooling injury and the rapid cooling injury. So how come a thermodynamicist is working in this field? In thermodynamics, we predict things and understand things like the phase diagram. This is a picture of the phase diagram of glycerol and water solution. So on this y-axis, we have temperature, and on the x-axis, we have glycerol mole fraction. So 0 glycerol means all water, and over here, 1 glycerol means all glycerol. So if I have any composition of water and glycerol and I'm at 20 degrees Celsius up here, my solution is liquid. But if I cool my solution down to a lower temperature, eventually I'm going to have ice freezing, or for highly concentrated solutions, I'm going to have glycerol precipitating out. That's what this phase diagram tells us. So if I take some cells and I start out with, let's say, 10% glycerol in a water solution and I cool them down, if I go below the freezing point for that solution, the solution is going to break into 2 pieces. Some is pure water ice, and some is a solution with a more concentrated glycerol solution. And as I cool it down, I get more and more ice and more concentrated glycerol solution. So I would want to predict this freezing line. What's the freezing temperature depending on how much glycerol I have in the solution? Okay, what about vitrifying tissues? So in vitrifying tissues, we also have to worry about the glass transition temperature. So now I could start with a very low concentration of a cryoprotectant, and then I would have to cool very fast to get all the way from this freezing temperature down to this glass transition temperature before ice even had a chance to form. And this cooling rate is so fast that we really can't practically do that for large pieces of tissue. On the other hand, we could start with a higher concentration of cryoprotectants. Then the gap between the freezing temperature and the glass transition temperature is not too much, and we can use a reasonable cooling rate. But now the problem is the solution itself might be toxic at a high concentration. So there are research groups working on how to cool and warm things fast. There are other research groups working on how to come up with multi-component solutions that are less toxic, but that vitrify. Our group's approach has been to add a little bit of cryoprotectant and cool it down, add a little bit more cryoprotectant, cool it down a little more, add a little bit more cryoprotectant, and cool it down even more. So this allows us to get enough cryoprotectants that we can vitrify, but we added them at lower and lower temperatures and we mitigated the toxicity. So for our approach also, we need to know this freezing line. How does freezing point depend on the concentration of the solution? So thermodynamics, we have an equation for chemical potential of water. It's equal to a function that depends on osmolality, which is a function of the concentrations or molalities of each of the solutes in there. So I could have salt in there, I could have glycerol as a cryoprotectant, I could have another cryoprotectant like dimethyl sulfoxide, and a function of these would show up in the chemical potential. So if I want to compute this freezing point line, I would say the freezing point happens when the chemical potential of water in the ice equals the chemical potential of water in the solution, and out would come an equation for freezing point that has this function osmolality in it. Another thing I might want to know in cryobiology is how much the cells get dehydrated in the slow cooling injury. I could say the chemical potential of water in the solution inside the cells equals the chemical potential of water in the solution outside the cells. And this is going to give me osmolality inside equals osmolality outside. So to predict this dehydration or the freezing point, I need to know this function osmolality. So one thing that my group has added to cryobiology is we've proposed this function for osmolality. If you were in your lab and you measured osmolality of a solution with an osmometer, this first term would give you the ideal dilute solution, just adding the osmolality from one measurement to the osmolality of another measurement. But we proposed this more complicated equation, which takes into account all the interactions and non-idealities of concentrated solutions. So how good is this equation? Lucky for us, it turns out to be fantastically fairly accurate. So this is the freezing temperature of a water solution with dimethyl sulfoxide and glycerol, two common cryoprotectants. The dots are the experiments, and the blue line is the prediction of the freezing point using our equation for osmolality. Here's another prediction for dimethyl sulfoxide and salt solutions. And we've actually made these predictions for all sorts of things like mixtures of proteins, sugars, salts, cryoprotectants, and all of them are very, very accurate. So that's very lucky for us because it allowed us to take cryobiology to a very accurate computational things that we can do. And over the years, our group and other research groups have made many improvements, applying this to electrolyte solutions, lots of different cryoprotectants and protein solutions, even the inside of living cells, cytoplasm, it's a pretty good predictor. And finally, in 2023, Hikmat Bidiamanov has even added a little bit more theory and applied this to all sorts of complicated salt solutions. So the point of this part is that we know how to predict freezing point of a solution of mixtures of molecules. And this work was very well received, and actually it's commonly used by many researchers in cryobiology, and very exciting for me. 10 years after this paper was published, it was selected to be republished in a special issue honoring the birthday of Marie Curie. So that was very exciting for us. Okay, so that's the basic thermodynamic thing we added to cryobiology. How can we use this information to make progress in cryopreserving individual cells in the presence of ice or in ice-free vitrification of tissues? So first, I'll give some examples of classical cryopreservation. One is going to be improvements to cryopreservation of cells in suspension, and another is going to be cryopreservation of cells in layers. So I first want to mention this tissue of cornea. So this is the clear tissue on the outside of your eye that covers the pupil and lets you see through it. It's made up of a stromal layer with some keratocytes, an epithelial outside layer with some epithelial cells, but the really important layer is this single layer of endothelial cells on the inside. And this single layer of endothelial cells controls the water balance of your cornea. And so if you get losing some of these endothelial cells to disease or damage, you'll lose the water balance. Your cornea will get cloudy, and disease or loss of endothelial cells is one of the main reasons for corneal blindness and needing a cornea transplant. Actually, cornea is one of the most widely transplanted tissues. Currently, we don't have a good method to cryopreserve it, so it has to be transplanted fresh, which means that it can be only stored at refrigerator temperatures for a limited time, like 28 days, after which it would outdate. So one of the reasons also where corneas are difficult to cryopreserve is that this endothelial layer is very sensitive to cryo damage. And there's a couple reasons. One is because the cells themselves are actually challenging to cryopreserve, but also a layer of cells behaves differently than cells in suspension. So we're really interested in cryopreserving these endothelial cells, both as individual cells and in monolayers. Well, here's a picture of a blood vessel. It also has a lining of a single layer of endothelial cells. And the cryosensitivity of these endothelial cells is a reason why it's difficult to cryopreserve blood vessels as well. So this is the tool that we use, an experimental procedure called graded freezing. And this was proposed by Farrington and McGann in 1976, but it's a tool that we're using every day now. If we start out with some cells in suspension, we first cool them down to an intermediate temperature like -5 degrees Celsius and nucleate some ice, wait a few minutes for the latent heat of fusion to go away. And then we slow cool these cells. At different temperatures along the cooling profile, we'll warm them up and assess their viability. These are the direct thaws, and they give us information about slow cooling injury along this path. We also take some other cells, and at points along the path, we plunge them into liquid nitrogen, which is very rapid cooling. We store these cells and warm them back up and assess them, and we call these the plunge thaw samples. These tell us information about rapid cooling injury. So from doing this experiment, we can look at the patterns that result in the results, and we can use them to tell the difference between the slow cooling solute effects injury and the rapid cooling intracellular ice injury. So here's an example of what this looks like. If you look at this blue dotted line, this is what happens if you direct thaw some cells with no cryoprotectants. So if you took cells and you cool them down to -5 degrees Celsius, nucleate ice, and then warm them back up, actually they're all fine. They almost all survive. But if you cooled them down to -15, then you would have had only about half the cells are surviving that. So there's some slow cooling injury. And we know if we see some solute effect slow cooling injury, we should add some permeating cryoprotectant. So the green lines are what happens if we add 10% dimethyl sulfoxide. So now there is no slow cooling injury. In fact, if we have no slow cooling injury, it means we can actually probably get away with reducing the amount of permeating cryoprotectants. On the other hand, if you look at these plunge thaw results, if I had no cryoprotectant, none of the cells survive being plunged into liquid nitrogen, this blue solid line at the bottom. But if I have 10% dimethyl sulfoxide, I actually can get some cells surviving the plunge thaw. So plunge thaw with 10% dimethyl sulfoxide, I have about 70% of the cells surviving. But the difference between the direct thaw and the plunge thaw gives me information about intracellular ice rapid cooling injury. And in order to mitigate that injury, I should add some non-permeating cryoprotectants. So if you cryopreserve these human umbilical vein endothelial cells in suspension with 10% dimethyl sulfoxide, which is actually what the commercial distributors are doing right now, when you get that vial of cells that you purchase, only about 70% of them are alive. In our lab, being very careful with everything controlled, we have about 73% of the cells surviving. But this pattern of this experiment tells us we can actually do better if we reduce a little bit the permeating cryoprotectant and add a little bit the non-permeating cryoprotectant. So what we did is we ended up optimizing this procedure, and we used 5% dimethyl sulfoxide, 6% hydroxyethyl starch. We equilibrate those on ice for 15 minutes. We cool to -5 degrees Celsius, nucleate ice, hold for 3 minutes, cool at 1 degree per minute to -35 degrees Celsius if we're doing vascular endothelial cells, or to -45 degrees Celsius if we're doing corneal endothelial cells. And we've done these experiments for pig corneal endothelial cells and human corneal endothelial cells, and then plunge them into liquid nitrogen. When we warm them back up, the vascular endothelial cells now have a viability of 94% of fresh control. Remember, the current procedure before this work was 70%, so that's a big improvement. For pig corneal endothelial cells, we have 91% viability of fresh control. And for human corneal endothelial cells, 93% viability compared to fresh control. So that's great. And we can also look at some of the functions by taking those cryopreserved cells, warming them up, culturing them for 7 days, and just seeing if they express some of the enzymes and tight junction markers, etc., of healthy cells. And you can compare these from fresh and frozen, and they do all the same things. This is for pig corneal endothelial cells. A function for the vascular endothelial cells is that one thing that vascular endothelial cells do is they're involved in forming new blood vessels. And so if you put them on the right matrix, which is matrigel, which has some signaling things, it's going to induce healthy cells to form new tubes. So here are some fresh endothelial cells forming these tubes, and here are some cryopreserved cells forming these tubes. So we really improved cryopreserving endothelial cells in suspension. Here's just another example from our group about using graded freezing to improve the preservation of pancreatic islet cells. These cells we cryopreserved in 10% dimethyl sulfoxide and 6% hydroxyethyl starch. And our collaborators at Stanford University use single-cell RNA sequencing, and our collaborators at the University of Alberta use single-cell electrophysiology experiments to show the presence in the surviving cells after cryopreservation of all the major pancreatic islet cell types, alpha, beta, delta, and PP cells, and also showing that after cryopreservation, the key electrophysiological parameters were comparable to those of fresh cells, and just learning a little bit about the differences. Like here I'm saying it's all good, but there are some differences between the fresh and cryopreserved cells for these pancreatic islet cells, and actually we see the involvement of a gene related to ferritin, telling us something about the mechanism of cryo damage. For this work, we have some other funders in addition to the ones on my front page. Well, another example is using graded freezing to cryopreserve cells in glycerol. So this is graded freezing for some cardiac myoblasts, another type of cell line. And traditionally, these cells are cryopreserved in 5% dimethyl sulfoxide. That's the blue line. We could do better with 5% dimethyl sulfoxide and 6% hydroxyethyl starch. That's the red line. But there are some reasons why people would like a procedure that doesn't use dimethyl sulfoxide. So glycerol is another cryoprotectant. It's very non-toxic. It might be desirable, but it's very slow to permeate cells. So we use this graded freezing to come up with good loading procedures for glycerol. So here we loaded the glycerol at room temperature for 2 hours. That's quite long, but we're able to cryopreserve these cardiac myoblasts with 82% surviving the glycerol procedure. It's not as much as the 91% with our best procedure with dimethyl sulfoxide and hydroxyethyl starch, but you could use this glycerol in a case where you're happy with 82%. Remember that the commercially available ones are only at 70%, so this is still very good. Here's another example of a different cell type. These are some brain microvascular endothelial cells, and the glycerol procedure is almost as good as the dimethyl sulfoxide and hydroxyethyl starch procedure. So up to now, I just showed a few examples from our group of using this graded freezing to do a better job and improve the cryopreservation of cells in suspension. I'm just reminding you, this is the picture I had before for human umbilical vein endothelial cells in suspension. But what if we now grew those cells in a monolayer and we tried to cryopreserve that monolayer? The pattern of response is very different for these same cells in a monolayer. So if you look at this plunge thaw with 10% dimethyl sulfoxide, 70% of cells in suspension survive, but only 10% of cells in a monolayer survive. So what different is going on for these attached cells? Well, one of the things that's going on is ice growth through gap junctions. So if I have cells in a monolayer, many of those cells express gap junctions, which is a little protein hole that lets ice more easily grow from one cell to the other and get some enhanced ice propagation. Again, I can come up with my chemical potential of water in the ice equals chemical potential of water in the solution. And because of this curved interface and surface tension between ice and water, the pressure in the ice is greater than the pressure in the solution by an amount related to the ice-water surface tension and the radius of the pore. In case you're wondering if ice does grow like a little spherical interface like that through a narrow pore, here's a picture that my collaborator took in of ice growing in a glass capillary. So it's a 20 micron glass capillary. The clear part is ice here. The colored part is liquid with a bit of potassium permanganate, just so you can see it. And you'll see the interface is indeed part of a sphere. So we can combine those two equations. Now we got another equation for freezing point. It still has the part that depends on the solution composition and osmolality, but now the freezing point also depends on the ice-water surface tension and the radius of the pore. So now I want to discuss a couple experiments about that. It's kind of on a side thing from cryobiology, but I think it's interesting. My collaborator here, Braslavsky, in his lab, they did some interesting experiments where they put some antifreeze proteins on ice, and then they looked at the temperature at which ice grows between these pores, and they did that. They put the proteins apart by ranged between 7 to 35 nanometers apart. In my collaborator Loxley McGann and my kind of joint lab, actually Jason Acker in his PhD thesis, he measured the ice propagation through gap junctions, and these gap junctions have diameters of about 1.5 nanometers. Another collaborator, Carlson, in his lab measured ice growth through these tight junctions, the junctions between cells. These have a diameter of 0.7 nanometers. That is so small, actually. That's like 2.4 water molecules across. It's shocking and surprising that we would think thermodynamics could ever be applied at this scale. But from these 3 different labs for 3 different experiments with 3 different proteins across pore sizes, we find that this curvature effect fit to these experiments gives out 1 protein-water-ice contact angle. So it's kind of shocking, but this curvature effect describes ice growth through 1 nanometer pores. And this is actually the limit, the smallest size to which this kind of surface thermodynamics has been shown to be accurate. And this is just an invited perspective article that I wrote in Journal of Chemical Physics on thermodynamics at the nanoscale and some reasons why that is so. So that's kind of an interesting thermodynamics outcome of this story. Okay, back to actually freezing some monolayers. So again, we used our graded freezing to examine the role in monolayer cryopreservation of the cryoprotectant. So we're using 5% dimethyl sulfoxide and 6% hydroxyethyl starch. But now we're also investigating reducing this intercellular ice propagation. We used a substrate. Instead of culturing the cells on glass, we cultured them on a vinyl polymer called Rinzo that has a coefficient of thermal expansion matched to ice. So this means as the cells and their substrate are cooling down with ice outside, the substrate and the ice are shrinking at the same rate, so you don't have any extra stress on cells, hypothesizing that this extra stress can also open up some membrane pores and make the ice propagation even easier. Also, we added chondroitin sulfate. So chondroitin sulfate acts like an antioxidant, but it also acts like a membrane stabilizer. So here is the viability of some cells cultured on glass with no cryoprotectant and cooled at 1 degree per minute. The little blue bar here, almost none of them survive. On the other hand, even with no cryoprotectant, if you put them on Rinzo plastic, 40% of them survive the cryopreservation. And here it shows if you culture them on glass, even with the very best cryoprotectant, 5% dimethyl sulfoxide, 6% hydroxyethyl starch, still only less than 20% of the cells survive. But if you do that on Rinzo, tons of them survive. So this was a new idea in cryobiology, using a substrate with the same coefficient of thermal expansion as ice. And it really allowed us to cryopreserve these monolayers with a huge improvement in viability. And if we add chondroitin sulfate, actually we even do a little bit better. So this is just a picture of some cells. They're pig corneal endothelial cells. If you grow them on a monolayer and just plunge them into liquid nitrogen with no cryoprotectant, then warm them back up and stain them with a stain that stains cells with damaged membranes red, you'll see that all of the cells on this are red. They're all dead, and there's even some black patches in between where the cells have been lost. On the other hand, if you use our new procedure, which is with Rinzel cover slips, 5% dimethyl sulfoxide, 6% hydroxyethyl starch, and 2% chondroitin sulfate, when you warm those cells back up and stain them with a stain that's red, you see a few red cells here with damaged membranes, but the rest are all green with intact membranes, 91% viability for these vascular endothelial cells, and 92% viability for these pig corneal endothelial cells. So we can cryopreserve monolayers with excellent cell viability using our new procedure. And we've applied that to some brain microvascular endothelial cells, also another kind of cell in the brain, astrocytes. And recently, this 2024 paper even shows that after warming these cells back up, you can actually keep them in culture for a day and they're still perfectly healthy. So this is a huge improvement in monolayer cryopreservation. So next, those two examples were cryopreserving things in the presence of ice. Now we want to talk about an example where there's no ice inside of cells or outside of cells. It's vitrification. So again, the example is going to be cryopreserving articular cartilage. So this is a picture of a knee. Articular cartilage is this white tissue on the ends of articulating bones in articulating joints that lets them slide smoothly over one another. It's a living tissue with some chondrocytes. As this tissue gets damaged, the chondrocytes repair it. If you get a small damage to this articular cartilage, then the cells will heal it, it will grow in. If you get a larger piece of damage, the cells may not be able to grow in that new cartilage, and you may need some surgical intervention. For even larger pieces of damage, we know that transplanting articular cartilage, so transplanting a larger portion of articular cartilage with some underlying bone, is an important treatment. But this treatment has to be done using fresh tissue because articular cartilage was one of the tissues that we did not have a good procedure for cryopreserving. But for almost 30 years, myself and my collaborators, Loxley McGann and orthopedic surgeon Nader Jama, we worked to preserve intact human articular cartilage on bone for use in transplantation. How are we going to do that? We're going to add some cryoprotectants, cool fast, and vitrify. Now, why is this a hard problem? If we take some dimethyl sulfoxide, 6 molar dimethyl sulfoxide would be enough to vitrify. If we put some cells in that, in 15 minutes, they're almost all dead from toxicity. On the other hand, if we look at how long it takes for dimethyl sulfoxide to diffuse into cartilage, this is a picture of concentration versus the place inside the cartilage. So over here at 0 is the bone. Over here at 2 is the solution. So at time 0, we have lots of cryoprotectant in the solution, but no cryoprotectant in deep. After 1 minute, some of the surface cartilage now has higher concentrations of cryoprotectant, but in deep, there's still no cryoprotectant. Even after 1 hour near the bone, it only got to 3 molar when the surrounding solution is at 6 molar. So this is why this is a hard problem. It takes hours to diffuse in enough cryoprotectant, and it takes 15 minutes for that cryoprotectant to kill the cells. So that's a hard problem. And we use some mathematical modeling to solve this problem. So ice formation has to be prevented inside the cartilage matrix. We need to diffuse in a vitrifiable amount before the toxicity limits of the cells are reached. And actually, I mean, it's been 27 years for me, more than 30 for Nodder and Loxley. So we used up lots and lots of grad students doing diffusion models, vitrifiability models, toxicity models. I'm just going to mention the diffusion biomechanical model today. So this is cartilage. It's basically a solid with some water and ideal solution on the bone side and the bathing side. When you first put it in this concentrated cryoprotectant solution, water is going to flow out, cryoprotectant is going to flow in. While the water is flowing out, the cartilage is shrinking. So this is like a moving domain problem. And eventually the cryoprotectant is going to build up inside this cartilage in a spatially inhomogeneous way. So, and then the other thing is that this cartilage acts a bit like a spring that is resisting the movement of water and cryoprotectant. So this is what that model looks like. It looks a little complicated. The blue parts are the part that we added. And again, we're using our osmotic virial equation. You see these osmotic virial coefficients showing up in the chemical potentials of water and cryoprotectant that are involved in this non-dilute transport. We actually know everything in this model except for these 4 orange things. And we got those 4 orange things by fitting some experiments with this mathematical model. This math model is surprisingly accurate. When we fit for these orange things, one of the things we get out is the cartilage stiffness. So this is just saying how stiff is the spring? How much does the cartilage resist shrinking? How much pressure difference is needed for the cartilage to shrink? And when we back out that stiffness from our transport model, it actually measures force, it matches force measurements of cartilage stiffness. So taking that diffusion model together with vitrifiability model, together with toxicity model, and lots of biological experiments over more than 20 years, we came to this procedure for cryopreserving articular cartilage. We take the cartilage and put it in 6 molar dimethyl sulfoxide for 1 hour and 30 minutes at 0 degrees Celsius. Then we take it out from that solution and put it in 6 molar glycerol and 2.44 molar dimethyl sulfoxide. Keep it there for 3 hours and 40 minutes. Then we take it out and put it at -10 degrees Celsius and load some propylene glycol. Take it out and put it at -15 degrees Celsius and load some 6 molar ethylene glycol. And finally end up with this final solution, then plunge it into liquid nitrogen. So we've used the idea that was put forward by Greg Fahey and others that adding a bunch of different cryoprotectants, you can get something vitrifiable, but it's not as toxic as if you use one cryoprotectant. It's also got the idea that has been championed by PEng and others that we can add some cryoprotectants that successfully lower temperatures to mitigate a bit of the toxicity. But the point is, you will never guess this procedure by trial and error. You need some math models and you need to compute it. And after we cryopreserve this cartilage and warm it back up, here is some human articular cartilage. This is what the fresh cartilage looks like. This is what after being vitrified and warmed back up looks like, with 70% of the cells alive and green. So we patented that procedure and we are very excited about it. This paper was first published in 2012. Actually, this takes a long time to load the cryoprotectants. This procedure is more than 1 shift time in a tissue bank, and we've been doing a lot of research since then in kind of optimizing things towards more practical usage in the tissue bank. Here's an example from a paper 2021. We want to cryopreserve small cubes of cartilage. This is pig cartilage and human cartilage. And we have 2 different procedures. One of them involves loading ethylene glycol and dimethyl sulfoxide. At the end of the second loading step, you'll see the ethylene glycol concentration across the tissue from the bone to the surface, or from this is from the middle of the cartilage, sorry, to the surface, no bone in these little cubes. You'll see the freezing point across the tissue. You'll see our predictions of vitrifiability everywhere in the tissue. The amount of cryoprotectant has exceeded our vitrifiability threshold. So we predict this will be a good cryoprotectant protocol, and guess what? It is. If you warm some cells up after cryopreserving them, they have comparable viability to your fresh control. In fact, even if you keep them for 6 months in liquid nitrogen and warm them up, they have very much viability. And we showed that for pig and human cells, and also lots of functional assays on these cells in this Nature Partner Journal, Regen Med. So this was a really exciting paper for me because we're actually at the stage where we can just get a new product that a tissue bank wants to cryopreserve, do our math and predict exactly how you should cryopreserve it, and then we go in the lab and try it out, and it works really great. And the type of research we've been doing since then is some improvements towards actual practical tissue banking in human clinical tissue banks. So effect of adding sucrose into our vitrification solutions, using clinical grade chondroitin sulfate and antioxidants, and also after you vitrify it, showing that when we warm it up at the tissue bank, we can actually store it for a week before distributing it. So that was a quick journey through some of the research in our group. Based on improved thermodynamic models combined with experiments, we have achieved predictive mathematical modeling of cryobiology. And we were able to use that to improve the slow cooling cryopreservation of endothelial cells. So actually from about 70% alive to now more than 90% alive. Also, I showed you a few other examples, lots of other cell types that we have published papers on. We also came up with a way to cryopreserve endothelial monolayers. If you just use 10% dimethyl sulfoxide, only 10% of cells survive, but in our current cryopreservation procedure, more than 90% of cells survive being cryopreserved in monolayers, and we showed that for lots of different types of endothelial cells, like vascular endothelial cells and corneal endothelial cells. And also we have cryopreserved by vitrification human articular cartilage on and off bone with high post-thaw chondrocyte viability and function. And well, you've heard it said that a picture says 1,000 words. I like to say that an equation draws 1,000 pictures.
Maybe I'll start with a general question, Janet. So you presented two scenarios where we're using slow cooling 1 degree per minute and then fast cooling, tens, hundreds of degrees per minute. Why aren't we working in the middle?
Okay, that's a good question. When we say slow cooling and rapid cooling, really we should have said cryopreserving cells in the presence of extracellular ice and cryopreserving cells with no ice at all. And the no ice at all vitrification requires very high cooling rates, but it's also very challenging for a number of reasons. So we only really use that vitrification if for some reason the slow cooling ice present one did not work. So we're kind of aiming towards vitrification for the larger tissues and for eventually organs. But for cells in suspension, they're perfectly happy to have ice outside of them. They just don't want to have ice inside of them. The strategy for the ones with ice outside is you need to cool slowly through this intermediate zone in order that water has enough time to leave the cell and freeze outside and not freeze inside. So it's kind of like if you can have ice outside, the strategy is slow cooling. If you can't have ice outside, the strategy is vitrification. So it's two different pieces of physics, and an intermediate cooling rate doesn't really solve one of those two problems.
Awesome. We do have a question in the chat. Lothar is asking, can you apply these equations for immune cell freezing? And maybe I'll expand it to, have you applied these equations for immune cell freezing?
So we have not, I'm trying to think, have we done we've done other things like measure all the parameters of some immune cells, but we have not actually done I have not personally done graded freezing experiments for these immune cells, but these principles apply to all cells in suspension. So, we have looked at a wide range of cells in suspension. If you want to cryopreserve cells in suspension, these are the strategies that you would use to apply all the cryobiology we know right now. The problem with immune cells is they do have some extra biology things that is what makes them challenging to cryopreserve in some kind of activated processes. So I'm not saying for sure that you would solve all the problems of immune cells, but if you want to apply everything we know about cryobiology right now to immune cells, you could at least come up with your best possible slow cooling procedure. Fernanda Fonseca has a question here. What about the thawing conditions for different freezing conditions? Are you modeling this also? And I just want to say, okay, this is the problem I have when I give a talk. I never try to give a talk of one journal paper. I get excited and I want to give a talk of 50 journal papers. So necessarily I have to skip a lot of parts of the journal papers. But of course, when we say optimize, like if you go and read each individual journal paper, the types of things we've optimized are the actual whole cooling profile. So it's like what temperature we nucleate ice, how long we hold it before we start the slow cooling rate, the rate at which we plunge the cells into liquid nitrogen, and warming conditions, and cryoprotectant adding and removal conditions. One of the things about ice, when you have the ice present outside of cells, they're actually not too sensitive to the warming rate. So as long as you have ice outside of cells, which we nucleated ice, so we know we have it outside of cells, typically people know you can't just warm them on the bench, but if you put them in a 37-degree water bath, it's warm enough. And so for the slow cooling in presence of ice, we really don't have to worry about the warming rate. Like you can say it plays a role, but it really doesn't play too much of a role, long as it's fast enough. For vitrification, warming rate is most of the game. So for the vitrification, we really have to care about warming rate because we don't want any ice to form on the way back up. So some groups are really working on this nano warming and fast warming for vitrification. Our group worked in a different way, that we used the thermodynamics to find a complicated way to get more cryoprotectant. Because we have more cryoprotectant, ours are not too sensitive to the warming rate either. And so we can take a whole chunk of tissue, put it in a big tube, and warm it in a water bath, and we still have pretty good vitrified tissue results. So we have not focused on using warming rate as a strategy. We've worked on using knowledge of chemical thermodynamics as a strategy, but there are people working on the warming rate.
Thank you, Janet.
One final question, Lori.
Thank you to Janet. I don't think I can, sorry, I'm having trouble with my controls here.
Yep.
I can't turn my camera on for some reason. Oh, there we go. Janet, that was really so exciting. It's so exciting to see this stuff and just pie in the sky to end, maybe. We're hearing about shipping organs, worldwide and changing the way we allocate organs for transplantation and so on. Care to speculate?
That's a great question, because I would say if you asked me that, but if you did ask me the question, as a writer in the Proceedings of National Academy of Science did ask me 15 years ago, I would say it's never going to happen, cryopreserving organs. I was very saying that is never going to happen. Too hard science problem, But honestly, in the last 10 to 15 years, a lot of research progress has been made. And I'm a person who changed my mind. And I'm actually part of the board of the advisory board of the Organ Preservation Alliance of the United States. So I think there's a lot of challenges to be solved for cryopreserving organs, but people are working on all those individual challenges, and we know what those individual challenges are. I think our work on we can vitrify a whole knee, okay? We can warm it up and have more than 70% of the cartilage cells surviving at the end, Okay, like a whole knee is a pretty big bag of something to vitrify. So we can do that. I think that's one of the largest things that anybody has ever cryopreserved. An organ is a far, far way from that, but honestly, we can see it from here. So it's a very exciting time in cryopreservation. There's a really big movement in the United States where the National Science Foundation has put a lot of funding into these different groups working towards organ preservation. I don't know. I tend to say my work is on cells and tissues, but I think organ preservation will happen in the future.
Well, that's a good response. I can see it from here, I think is what you said. I can see it from here. That's great. Thank you.
Awesome. Thanks, Janet. Thanks everyone for attending. Just a few final comments before we close. There are a couple other comments, Janet, coming in on the feed. I'm not sure we can capture those and you can look at them later or respond. So this is the last seminar for the 2023-2024 season. We'll be back in September with the ATI Research Symposium. Thank you for all of our in-person and virtual speakers that have presented throughout the series. We're always interested to hear from you about your favorite speakers and subjects to be covered in the seminar series. So please contact us at transplant@ualberta.ca. Let us know future topics or speakers that you would like to have featured in this future seminar forum. But also please visit the ATI website as we will update it with the dates and speakers for 2024 and 2025. And with that, I will close and look forward to seeing many of you on May 15th at the ATI Research Day.